The foundation for healthy flowers, trees and vegetables is healthy and high-quality propagating material. Seeds, cutting and young plants can carry harmful viruses, bacteria, fungi or other organisms that can be invisible to the naked eye. These pathogens can cause problems later on during the cultivation process. Naktuinbouw has the knowledge and advanced equipment necessary to test propagating material in order to safeguard the quality of propagating material. Part of the laboratories at Naktuinbouw is dedicated to routine testing of the health of seeds and plant material. Naktuinbouw conducts laboratory tests to see if this propagating material is free of diseases. Focus is on large-scale testing on bacteria, fungi and viruses. Naktuinbouw inspectors and individual companies send samples, which can be for instance seeds, leaves or other parts of a plant, to Naktuinbouw Laboratories. After registration of the samples, these samples enter the testing process anonymously. Often a pre-treatment is required to make the sample suitable for testing. It is ground, squeezed or mixed, and a buffer is added to ensure optimal test conditions. To be able to detect fungi or bacteria, samples are incubated on specific growing media. After a specified period, the media are analyzed for the presence of the pathogens of interest. Naktuinbouw uses a variety of testing methods to detect viruses. A widely used test for plant material or seed is the ELISA method. The principle behind this diagnostic tool is the use of an antibody that will bind to a specific protein of the pathogen you want to identify. The antibody does not bind the other proteins. A change in color indicates that binding has taken place and that the virus is actually present. It is a reliable and relatively inexpensive technique that gives results within 24 to 48 hours. The PCR method is a more sensitive and specific diagnostic tool. With this method, material is tested at the DNA level. Pathogens can be detected in seeds, plant material, soil and water. Heat treatment can be applied to produce virus-free plant material for woody crops like, for example, apple and pear. Due to the enhanced temperature, growth of the plant is increased. The viruses cannot keep up with the speed of growth and the top tip of the plant becomes free of viruses. This tip is then grafted onto a virus-free rootstock. A number of subsequent tests can be applied to prove the virus-free status of the plant. Naktuinbouw can also apply specific bioassays, where a leaf or a piece of tissue from the plant to be tested is grafted on woody or herbaceous test plants. These so-called indicator plants are assessed during their development to see whether disease symptoms will develop. This broad-spectrum test allows to detect a broad range of pathogens like viruses, even those who are currently unknown. Naktuinbouw also performs routine testing to detect nematodes in soil or plant material. Nematodes are tiny, worm-like organisms that can cause serious damage to plants. Soil samples are rinsed with water. The water flow that contains the nematodes is then collected and examined. When plant material is tested, the material can be separated from the nematodes using a centrifuge or a misting device. Afterwards, isolated nematodes are examined. Another section of the Naktuinbouw Laboratories is equipped to conduct tests on seed quality. The most important tests on seeds are for purity, germination power, moisture content and a usable plants test. Purity determination looks at the extent to which a seed sample is free of impurities. Once the sample has been weighed, the impurities such as plant residues, sand or soil are separated from the seed and are individually weighed. This indicates how pure the sample is. Sometimes other seeds are discovered in the samples. These seeds can be identified by comparison to the Naktuinbouw seed collection and the online seed atlas. In germination rate testing, Naktuinbouw examines the percentage of seeds that produce a complete seedling after a certain number of days spent on a germination table or in a germination cabinet under perfect growing conditions. Naktuinbouw is accredited by the International Seed Testing Association to perform the purity determination and germination rate test and is able to issue the international ISTA certificates. Since the germination power cannot always be translated to practice, a usable plant test has been developed in cooperation with the sector. After a prolonged period of germination, seedlings are assessed for the presence of well-shaped seedlings with first leaves. The percentage of usable plants gives the propagator or grower a more practice-related indication of the quality of a certain lot of seeds. Sometimes a plant may show symptoms of a disease that cannot be identified. These plants can be examined in the Diagnostic Laboratory of Naktuinbouw. Plant material from all over the world is sent to the Diagnostics Laboratory. After an initial diagnosis, the expert decides which pathogens to test for. Pre-treated material is plated out on a selective growth medium and, after a specified incubation time, examined. Photographs of the material are stored in a photo database. Lab technicians discuss the results of the tests and make a diagnosis. In special cases, the cause of the problem is discussed with the research and development team. The customer is informed about the diagnosis so that the appropriate measures can be taken. Researchers at Research and Development work on the development of new and improved tests, for example when new pathogens are found. The R&D team has a continued focus to improve the routine tests. They also look at ways of translating new and often expensive scientific tests into methods that can be applied on a large scale for routine testing. When new tests are being developed, the pathogen collection of Naktuinbouw is used. This collection contains pathogens that were once isolated from infected plant material. Naktuinbouw also has a unique collection of plants infected with pathogens with particular viruses. These collections are essential in the development of new tests. The collections are also used for the validation of the test protocols. In other words, to determine whether a test meets its specifications. As well as developing and improving tests, the team also supports the other disciplines at Naktuinbouw. For instance, the inspection department can be assisted by providing background information about new pathogens. To support variety testing, R&D can determine the kinship of new and existing varieties at the DNA level. This information is used to select the most closely related varieties for comparison purposes during variety testing. But DNA testing can also be used to reveal infringements of plant breeders' rights and to clear up mistaken identity of plant varieties. The laboratories at Naktuinbouw closely cooperate on national and international levels with other inspection services, the National Reference Center of the Netherlands Plant Protection Organization, universities and research institutes. Naktuinbouw forms a bridge between science and practice and contributes to high quality and healthy propagating material in the horticultural sector. Naktuinbouw is dedicated to quality.